welcome to the Scientist.com and Lonza webinar titled Discover the Freshness of Frozen PBMCs, an assessment of cryopreservation impact on T-cell activation and phenotype. I'm Liam Sanyo from the events team here at Scientist.com, and I'll be your host today. We're very fortunate to be joined by Dr. Sarah Thacker, Dr. Katie Dunnick, and Dr. Orita Menezes. Today, Katie and Sarah and Orita will explore the impact of cryopreservation on PBMCs, on T-cell activation and phenotype. So Sarah assisted with the research and will be giving the presentation, and Katie was the lead researcher and will handle the Q&A supported by Orita. Sarah, thanks so much for joining us today, and the floor is yours whenever you're ready.
I'm excited to talk to you today about the effect of cryopreservation of PBMCs and the source of those PBMCs and the downstream impact of the on the cell populations and their function. So to give you a sense of what we'll be discussing, we have the background on PBMCs and their utility in research and development, followed by the study design for a fresh versus cryopreserved leukopak. And then we have the downstream functional impact on PBMCs. And finally, we'll conclude with key learnings from this investigation. So what are peripheral blood mononuclear cells, otherwise known as PBMCs? PBMCs are a mixed population of specialized immune cells, which are comprised of myeloid cells as well as lymphoid cells. And you can see in this schematic that there are many cell types downstream of these two populations. This is actually quite simplified for all of the cell types that are comprised in the PBMCs, but it's important to know that they play a very essential role in the innate and adaptive immune systems and are critical for protecting the body from infections. Additionally, upon interactions with pathogens, cytokines, or chemokines, PBMCs can fully mature into these specific T cells, dendritic cells, and other subpopulations. So PBMCs and downstream immune cells are essential for many key applications and research initiatives. This includes some of the things listed here on the slide, such as immunogenicity screening, immuno-oncology, CRISPR and STING testing, cell and gene therapy, humanized mice, drug clearance, and large quantity cell isolation. And you can see that from each of these different applications, there are specific subpopulations within the PBMCs that may be utilized. So it's very important to have good quality material so that you can be successful in your downstream work. So as I just mentioned, it's very important that the PBMC quality and the starting material of those PBMCs are of good quality to be successful. So a lot of times with researchers, they may acquire PBMCs through a fresh leukopak, where the blood sample is collected from a donor. A leukopak is acquired fresh, and then fresh PBMCs are isolated, which then in turn leads to downstream immune applications and isolation of specific immune subpopulations, and then the utilization of those subpopulations in various applications. The challenge with this approach is that this process can take quite a bit of time. If you think about the collection from acquiring the fresh leukopak to then the fresh isolation and then utilizing these cells in your downstream applications, all happening in the course of 24 to 48 hours, this is something that's not always feasible, and you may be challenged with getting the availability of the donors of interest and the timing of your choice. So it makes it challenging to predict and therefore downstream studies are difficult to plan. Complex applications with multiple donors as well as repeat donor studies may not lend well to the inconveniences of using fresh leukopaks and fresh PBMCs. So this led to the question is what is the impact of using cryopreserved leukopaks as the source of PBMCs on the downstream function and population of immune cells? And so here I present to you our study design to answer this question. So we collected leukopaks from 4 different donors. Their blood samples were then split into a fresh leukopak, as we may traditionally be familiar with, as well as a leukopak that was cryopreserved and then shipped to us as well. So then we took those fresh or cryopreserved leukopaks and isolated PBMCs and cryopreserved those PBMCs according to Lonza's protocols. And what I want to emphasize here is how we term these 2 different samples. So we either will refer to this as a fresh versus cryo preparation, or you may see it in the presentation as a single cryopreservation or a dual cryopreservation. And what that indicates is how many times the sample has been cryopreserved. So for the fresh leukopak and cryopreserving PBMCs from that isolation, it'll be a single cryopreservation. And from the cryopreserved leukopak that then we isolate and cryopreserve PBMCs, it'll be a dual cryopreservation. So downstream of doing these isolations, we assessed the PBMCs in their function and subpopulations in a variety of ways. So we started with looking at PBMC viability post-thaw. And from there, we either did immunophenotyping of the PBMC cell population via FACS, so flow cytometry. And this looks at a whole host of different subpopulations as indicated on the slide. And then we also, in parallel, looked at T-cell proliferation of these PBMCs using a CFSC dye as well as flow cytometry. And from those cultures, we took samples to also assess cytokine release using an ELA instrument. And with this, we looked at both pro-inflammatory cytokines as well as anti-inflammatory cytokines. So here are some initial results from the viability testing. So what you can see here is a combined donor. So of those 4 donors, we have this combined here in a single graph. And on the left-hand side, the dark purple is the single cryopreservation sample, and the light purple is the dual cryopreservation sample. And what you can see is that regardless of the number of times it was cryopreserved, both samples had viabilities well above 85%, which is very high, as the industry standard is greater than 70% for kind of a acceptable quality of PBMC. And what we can see also is that there is minimal difference between the single cryopreserved or the dual cryopreserved samples. And so this was the first indication that the cryopreservation of the leukopak did not impact that PBMC quality. So next, I can tell you a little more about the T-cell proliferation process. This starts as PBMCs were thawed and counted. Cells were then incubated with the cell trace CFSC cell proliferation dye for 20 minutes. And after stopping this reaction, the cells were resuspended in Lonza's ex vivo 15 serum-free media and plated in 3 different conditions. We first have here ex vivo 15 media only with the PBMCs. We also have ex vivo 15 media with the PBMCs and the addition of IL-2, which is a cytokine that supports the proliferation of the T cells that are within that culture. And finally, we have the ex vivo 15 media with the PBMCs, IL-2, along with CD3, CD28 dynabeads, which are added here to activate that proliferation of the T cells within the culture. So to give you a little more context on the CFSE dye and how it works to assess proliferation, we have the CFSE dye, which crosses the plasma membrane and covalently binds to all free amines on the surface and also inside of cells. And because of the covalent nature of this binding to the target cells, the CFSE dye will remain in those initial parent cells, but also be incorporated into daughter cells as proliferation occurs. And this dye can actually be visualized for up to 8 generations of proliferating cells, which allows for a very robust assay. So to continue, these cultures were done for 4 days. The cells were stained with live dead reagent for 30 minutes in the dark on ice, and they were fixed in 2% PFA and analyzed using flow cytometry. So just to show here, to make it clear to everyone who's not familiar with CFSE, you're actually going to have the highest level of CFSE signal at day 0, as indicated by this blue peak. It's most rightward shifted, which means it has high level of fluorescence. And then as the culture continues and the cells proliferate and divide, that fluorescent signal gets depleted. And so you have a leftward shift, which is indicated by this green peak, but a very broad one at that. If the cells are proliferating, there should be a high number of cells with just a lower level of fluorescence. So to indicate, and you'll see in this study and other studies like this example, that if you have a high level of proliferation from your cells, you're actually going to have a low level of CFSE signal. So here are the results from that T cell proliferation. And this is shown both as individual donors on the left as well as combined donors of that N of 4 in the middle. And I'll just focus you onto that combined donors. And we have here as our negative control, that media only without IL-2, and that is indicated by the dashed line as kind of our negative control, what we would see in terms of minimal proliferation. And then we compare that to the IL-2 media with the cells, as well as the IL-2 media with the beads, the CD3, CD28 beads in the blue bars. And so what you can see is, as expected, there's minimal proliferation with the just the IL-2 media only, as shown in those purple bars. And it really is very comparable amongst the single cryopreserved and dual cryopreserved samples. And then you see much greater proliferation. Again, this is a lower signal, but higher level of proliferation. And that's seen in those blue bars where these cells were treated with the beads. And so again, single versus dual, very comparable here, no significant difference, and therefore we feel that the cryopreserved leukopaks do not impact that downstream T cell proliferation of the PBMCs. So here is the cytokine assay that was done. This I can walk you through. The cells were cultured for 7 days, again in the 3 different conditions as I described previously. We collected samples on days 0, 2, and 7 to show the progression and how those cytokine levels may change over the course of the culture. And the samples were prepared as the manufacturer recommends on this ELA instrument and loaded into a cartridge and read to determine the cytokine levels for the 5 different pro-inflammatory cytokines as well as the 2 anti-inflammatory cytokines. And so here what you can see is that really, I know this is a lot of data, so bear with me, but you can see on the left-hand side of each of these graphs, there's an underscore that says single. So that is the single cryopreserved samples. On the right-hand side is dual. So that is for the dual cryopreserved samples. And you can see in the kind of blue bars, again, those are the beads treated bars. That is where you see the significant levels of cytokines across all of the cytokines that were tested. Really, with the media only and the IL-2 media, you see minimal cytokine production, which is to be expected. But when you have high levels of proliferation, as we just saw occur with those beads, you also see that higher level of cytokine production. It does vary across donors and maybe even across cytokines in terms of the specific measurable amounts of cytokines. However, we do consistently see higher levels with those activated T cells. And we also really don't see any trends that support single over dual. So really, I feel that, again, with the cytokine production, similar to the T cell proliferation, the dual cryopreservation did not inhibit cytokine production. And that is true for both the pro-inflammatory as well as the anti-inflammatory cytokines. So to continue, we wanted to do one more check with the cytokines. In the absence of IL-2, we wanted to make sure that the adding IL-2, as we did in the previous experiment, was not influencing the response and production of other cytokines, such as IL-2 as well as interferon gamma. So we went ahead and repeated this just now at 4 days to kind of be in line with our optimized T cell proliferation protocol. But we have the media only as well as the media plus the activation with the CD3, CD28 dynabeads. And we measured that at day 0, 2, and 4 and measured IL-2 and interferon gamma and kind of compared that back to the previous experiment to make sure there was no influence with that additional IL-2. Results show here that similar to the previous experiment, we have minimal levels of production from the media only, as indicated in the green bars. And once you add those CD3, CD28 dynabeads, you actually start to see an increase in a very similar way to the previous experiment. So it turns out that in this case, the addition of IL-2 was not impacting those levels. And more importantly, kind of looking at that single versus dual cryopreserved sample, you are seeing very little difference among those PBMCs. And so, again, the dual cryopreservation did not inhibit that cytokine production in this case. And then finally, we have the immunophenotyping of the immune cell production. And so to walk you through this process, we took the PBMCs, thawed and counted them, incubated with an IgG control first, as well as followed by the target antibodies. And then we stained with live dead to get an assessment of the viability of these cells during this assay, and fixed and read on our FACS cytometer. And so again, we're looking at a lot of different subpopulations of these cells that you would expect to see in PBMCs and to determine if cryopreservation had any impact on that subpopulation. And so here are the results from the study. Again, it's a lot of data to look at, so I may redirect your attention to the graph on the right-hand side, which is the combined donor data. And to show that really, if you look at the dark purple and light purple bars, which represent the fresh or cryopreserved samples, you are seeing very minimal difference among those samples, nothing significant in terms of the statistical analysis that was done. And so what we can say here is, again, that dual cryopreservation process did not alter those subpopulations. We are seeing, I think, throughout all of these assays, there is going to be some donor-to-donor variability, and that is certainly to be expected with primary cells. That's one of the reasons why we assessed 4 different donors. But I think that when you combine them together, the trends are maintained that we are not seeing that effect when you cryopreserve the PBMCs twice. So to conclude, the cryopreservation of leukopaks does not affect PBMC function, and all of the functional applications of the PBMCs were maintained with fresh or cryopreserved leukopaks. So to kind of recap, the PBMCs isolated from both fresh and cryopreserved leukopaks retained very high viability at greater than 85%. The T cell proliferative response is a bit donor dependent, as I mentioned, and as expected, but is not altered by cryopreservation. The cryopreservation does not change pro or anti-inflammatory cytokine secretion profiles. This is also true when you remove IL-2, where IL-2 and interferon gamma cytokine secretion can be variable and donor dependent, but is not altered by cryopreservation. And finally, PBMCs isolated from cryopreserved leukopaks have similar cell populations as fresh leukopaks. So in addition to those scientific conclusions and the fact that we can maintain function and cell population of PBMCs, these frozen offerings offer many conveniences as well. Researchers can now choose from fresh or cryopreserved leukopaks as well as previously isolated cryopreserved PBMCs for a ready-to-use product. So this allows for several things, such as donor selection to meet one standard of performance. So this can be done with various applications, as I've shown today, such as immunophenotyping, T-cell proliferation, cytokine release. And you can do that because you can choose one donor and go back to that donor if needed, or choose other donors and understand that donor-to-donor effect. Additionally, there's a convenient timing of studies. As I mentioned before, processing everything fresh is certainly not conducive for a standard workday and may sacrifice on the scope and convenience of the study. And additionally, you could have repeat donor studies. So you really don't always know when a donor would be available. And with fresh material, it may only be available once to you. So in the case of cryopreserved PBMCs, you can come back to those same donors and confirm your results. And finally, donor variation to allow for more in vivo relevance to a population. So for example, you can actually pre-screen in advance of running your studies and look at donor demographics such as race, medical history, genetics, BMI, gender, things of that nature, and really make sure you have the right population to understand your endpoints. So to conclude, cryopreserved leukopaks and PBMCs are a convenient, equivalent, and easy-to-use option. That cryopreservation of both the leukopak and the PBMCs does not impact the immunophenotype, cytokine profile, and T-cell proliferative features of PBMCs. Cryopreserved PBMCs can improve the reproducibility and reliability of immunological studies. And as I've mentioned, you can experiment on your own schedule. So you can order and be delivered cryopreserved leukopaks. You can store them for later use. And the same with PBMCs. You can buy several amps and have them stored and ready to use on your schedule to offer that convenience and ease. So with that, I'd like to acknowledge the many people that contributed to this webinar today. This includes several members listed from our R&D team, as well as several members of our marketing team. So thank you very much to everyone at Lonza for their participation and contributions.
Fantastic. Well, thank you, Sarah, for a really great presentation. And with that, we'll move right on to the Q&A. So I'd like to welcome Dr. Orita Menezes and Dr. Katie Dunnick to join the audio line here. And as a reminder, if you do have a question you'd like to ask, you could submit it using the Q&A tab. All right, so first question here. We'd like to know, how can cryopreserved PBMCs improve the reproducibility and reliability of my studies? Can you elaborate on that?
Yeah, certainly. And like Sarah mentioned in the presentation, the benefits of using cryopreserved PBMCs is really having consistent donor-to-donor understanding. So not only does it save you time, you don't have to use them the same day, but you can ensure that your studies are really consistent between that same donor pool.
Fantastic. Yeah, great answer. All right, next one. Can you speak to the time frame for processing the fresh leukopaks versus cryo leukopaks?
Yeah, certainly. So the fresh leukopaks we process the same day that we received them. They typically come in about within 24 hours of being isolated. And then we would freeze those PBMCs after isolation that same day. The cryopreserved leukopaks were taken from the same donor, cryopreserved, and then shipped to us. And we kept those cryopreserved until we were ready to process them. That was typically between 1 to 3 weeks. And then we would isolate those PBMCs and cryopreserve them the same day as well. So everything was, the cryopreserved gave us a lot more flexibility on when we had to do the PBMC isolations.
Right. Yeah, it makes sense. Thanks a lot. All right. Have any additional cell types been isolated and their relevant applications tested with the single or dual cryopreserved PBMCs?
Yeah, definitely. So again, the flexibility of having cryopreserved PBMCs means that we can use them at any point. And so we've actually been able to isolate subpopulations such as CD4 T cells, CD8 T cells, and dendritic cells. And we've used them for some downstream applications. We haven't noticed any differences between what we call dual cryopreserved and single cryopreserved cells.
All right, perfect. Good question here. Are you concerned about donor-to-donor variability?
There's always donor-to-donor variability anytime you're working with primary cells. That's why for this study, we specifically used 4 different donors and then compared them across all of the donors. I think as long as you understand that there's donor-to-donor variability and you take that into account in your studies, you can control for that. And again, using a cryopreserved product means that you can buy the same donor to eliminate that variability for your initial studies if you're interested.
Yeah, great points. All right. How can I determine what donor options are available at any given time?
I will leave that question to Orita.
So yes, so definitely we have a large inventory of HLA typed PBMCs. So because many times people want to make a selection based on, the age, gender, and especially HLA type. So if you reach out to us, we can give you access to whatever is available and how many amps there are already cryopreserved. So it actually makes it very useful that you can select knowing exactly what you want rather than being dependent on whatever donor comes through the day typically in hospital labs. So reach out to us and we will definitely give you access to what we have available already isolated and cryopreserved.
Perfect. And Rita, what would be the best way to reach out to you then?
Customer service at Lonza, and they will put them in touch with the right salesperson who can then also support based on where the customer is residing. But definitely we can make that information available at the end of the webinar where to reach us.
Yeah, for sure. All right. Another good question here. So when using CFSE dye, do you need to account for the drop in signal due to the dilution through cell proliferation?
Absolutely. So this is something that we learned in our studies, was that depending on how many times your cells divide, you will eventually completely lose the CFSE signal in your stimulated cells. And so you have to kind of do some optimization of this assay for your specific endpoint. And for us, 4 days was a really good time frame. 7 days was too long. We lost all of our CFSE signal, but that's the whole point of the CFSE, is that when your cells divide, it decreases in presence.
Excellent. Right. Yeah. Okay. Another question here. So what's the source of the antibodies used in phenotyping of the cell subpopulations in the PBMCs?
Our phenotyping panel, all the antibodies are from BD Biosciences.
Okay, perfect. Okay, yeah. Did you follow the vendor dilution recommendation for the antibodies for FACS, or did you have to titrate to find the optimal dilution?
Yeah, great question. So we were lucky that some of these had already been optimized for us by a different team. So we were able to use those. But typically in my experience, I've used mainly what the vendors recommend and then dilute as needed based on the results.
Right. Yeah, it makes a lot of sense. Okay, what is your preferred viability stain for FACS or PBMCs?
For this panel, we used a near IR dead cell kit from Invitrogen. It works really well with the panel that we created because it was so many different channels. But there's quite a few other dyes that are available. But for us, it was just a live dead fixable dye.
Okay, perfect. All right. Another question here. Somebody says it's not very clear what is the dual cryopreservation. Is it a freeze-thaw cycle in this case, or is the difference in the leukopak additives?
Yeah, great question. It is confusing. So when we say it's dual cryopreserved, what that means is that the leukopak itself was cryopreserved, and then the isolated PBMCs from that cryopreserved leukopak were then cryopreserved again. So the cells theoretically have gone through 2 cryopreservation steps versus our single cryopreserved come from a fresh leukopak, how most people standardly process, isolated and then cryopreserved only a single time. That's how we distinguish dual versus single.
Okay, perfect. Yeah, great answer. Okay, yeah, how can cryopreserved PBMCs, oh, I think the question just...
It was a repeat question, sorry.
Okay.
Yeah.
Okay, yeah, somebody says it's not, yeah, is there a difference in the average number of divisions between the fresh and dual preserved?
So we didn't measure directly the number of divisions. We actually just looked at the signal of CFSE and compared the signal intensity, and we did not see any significant difference between the 2 different populations.
Excellent. Okay, great. Okay, yeah, somebody said, I always see lower monocytes like CD3 or CD14 positive population in the cryopreserved PBMCs. Have you ever seen the same change?
So both of these cells were technically cryopreserved at least once for our phenotyping, and we didn't see any significant differences in the CD14s. In fact, the dual cryopreserved populations actually had slightly increased. It wasn't significant, and when all 4 donors were combined, they were pretty equivalent. So we haven't seen that effect, but it could just be donor specific.
Right, yeah, makes sense. All right, good question here. So T cells are very robust cells. Have you evaluated the effect of dual cryopreservation on monocytes, B cells, or NK cells?
I believe our R&D team has done some work on the NK cells. I don't know, Arita, if you want to take that question.
Yeah, so we have tested monocytes as well as NK cells, and kind of gone through the differentiation process for monocytes and NK cells, and we do not see an impact. So we can use these functionally quite as similarly as T cell. I mean, it doesn't have the impact even on NK cells and monocytes.
Okay, perfect. Great to hear. Okay, yeah. Have you tested the effect of I guess it's a similar question here. Have you tested the effect of cryopreservation on NK cell cytotoxic function and activation?
Yes, we have. In fact, we have a published protocol with that, with using cryopreserved NK cells and cryopreserved PBMCs, and we've been able to show the cytotoxic activity.
Perfect. All right. So, are you able to combine the CFSE dye so that the fluorescence duration increases?
I don't fully understand that question. You typically only do the CFSE dosing a single time and then look for the decrease in signal. I'm not certain if you can redose or not. That's not something that we did in this experiment.
Okay, excellent. All right, another one here. So I know you saw good viability when thawing cells, but did you see any differences in the percent cell recovery? Do you have any tips for improving the cell recovery when thawing?
So we didn't see any differences in post-recovery with our frozen dual cryopreserved or single cryopreserved. Typically, I would say if you're putting your cells into cryopreservation with good viability, our recoveries are typically quite well. So in terms of recovering, making sure that you have a good product that goes into the cryopreservation process is a really good tip. And then your centrifugation speeds are also going to be pretty important, making sure that it's fast enough to get your cells, but not too fast that it's going to kill them.
Right. Yeah, that makes sense. And I think we'll just have another question here. So are there any particular developments or things that are coming up in the next 5 years that you're particularly excited about?
So, I mean, definitely, I think the field of biologics is expanding as well as CAR, and we see a lot of interest in testing our PBMCs in a lot of the immunogenicity testing for biologics in ADCC assays, as well as because we have a broad suite of products, including nucleofection products, in terms of developing in the research phase testings that to modify genes and to make CAR NKs and CAR macrophages. So I think the field is very exciting right now, and it really has a lot of potential in a number of different applications.
Yeah, definitely. Katie, anything you wanted to add on to that?
No, I think Arita covered it really nicely. Yep.
Yeah, great. All right, so do you find that isolating from frozen leukopaks, you see lower yields or purity of cells as some that may have died during the cryopreservation or thawing?
So like Sarah demonstrated, we actually didn't see that. Our recoveries, our purity, our viability, everything was really consistent between both the fresh and the cryopreserved starting populations. So we didn't see that.
Okay, excellent. Yeah, have you compared the function of fresh PBMC populations?
In terms of fresh, meaning they were never cryopreserved, we have not done those studies to my knowledge. But what this study nicely shows is that at least cryopreservation of PBMCs either once or twice did not have any impacts on performance.
Excellent. All right. And I think in the interest of time, I think we'll maybe call it there, unless there's any other questions from the list either of you wanted to cover.
I think we're good.
All right, excellent. Well, yeah, thanks so much for all your great insights today in the Q&A. It's really been a pleasure having you with us.
Thank you for having us.
Thank you. Thanks.
Yeah, definitely.
Bye-bye.
Big thanks also to the audience for joining us today. So in closing, we hope you enjoyed this Scientist.com and Lonza webinar, and we'll see you again next time. Have a great day, everyone.